Assalamu alaikum. This is the physiology lecture number 9 in the respiratory module. It's about the transport of carbon dioxide by the blood. What you need to know about this lecture, or the objectives of this lectures, are: first, enumerate the forms by which carbon dioxide is transported by the blood. Second, you should know how to describe the mechanism by which carbon dioxide is transported by the blood. And you need to know what is the Haldane effect on the transport of carbon dioxide in the blood. Before going into the details of this lecture, you need to know that the major difference between diffusion of carbon dioxide and of oxygen is that carbon dioxide can diffuse about 20 times as rapid as that for the oxygen. And the pressure differences required to cause carbon dioxide diffusion are far less than the pressure differences required to cause oxygen diffusion. The carbon dioxide pressure gradient in the body are approximately the following. Please refer to the figure. You can see here in this figure that the pressure gradient for the carbon dioxide in the lung is just 5 millimeters of mercury, simply because the pressure for the carbon dioxide in the alveoli is 40 millimeter mercury, and the partial pressure of carbon dioxide in the pulmonary capillaries is 45 millimeter mercury. Therefore, the pressure gradient is just 5. And in the tissue, the partial pressure of carbon dioxide within the cells are 46, while that in the interstitial space outside the tissue is 45. That's to say, the pressure gradient at the cellular side is just 1 millimeter mercury, and that pressure is quite enough for the carbon dioxide to be transported from one area to the other. Please refer back to the figure and notice this important point. See how quick is the diffusion for the carbon dioxide between the blood and the alveolus. Where I put the arrows, you notice that the carbon dioxide has been transported from the blood to the alveoli, and the pressure inside the blood has been reduced from 45 millimeter of mercury to just about 40 millimeter of mercury, and the blood has just passed less than the half distance within the capillary. That's to say, the transport of the carbon dioxide is so fast that the column of the blood within the alveoli has been cleared from the carbon dioxide even before the blood has been passing half the way through the capillaries. Two factors affect the level of carbon dioxide concentration in the venous blood. First, the tissue perfusion level. A decrease in tissue perfusion, as in hypovolemic shock, such as what happens after bleeding, increases peripheral tissue carbon dioxide concentration from the normal value of 45 millimeter mercury to elevated levels even as high as 60 millimeter mercury, and vice versa. What we mean by vice versa, it means when there is an increase in tissue perfusion, the partial pressure for the carbon dioxide will be lowered and this is expected. For example, if we give a patient a blood transfusion after the bleeding, the partial pressure for the carbon dioxide in his venous blood will be lowered. The other factor that affects the partial pressure for the carbon dioxide is the level of the cellular metabolism. An increase in tissue metabolic rate greatly elevates the interstitial fluid partial pressure for the carbon dioxide at all rates of blood flow, whereas decreasing the metabolism causes the interstitial fluid partial pressure for carbon dioxide to fall. An example for that is what happens during exercise. In exercise, there's an increase in the metabolism of the skeletal muscle. And if we measure the partial pressure for the carbon dioxide in the venous blood leaving the exercising muscle, we will discover that the partial pressure for the carbon dioxide has been elevated, and it will go back to normal, or it will be lowered by the end of the exercise. And there's many other examples for that. For example, when there is an increase in blood pressure or in tachycardia, the venous blood leaving the myocardium will have higher levels for the carbon dioxide. Under normal resting condition, an average of 4 ml of carbon dioxide is transported from the tissue to the lung in each 100 ml of blood. Let me repeat what I have told you before about the oxygen. In that lecture about the transport of oxygen in the blood, we mentioned that every 100 ml of the arterial blood carries 5 ml of oxygen to the periphery or to the tissue, and then back to the lung, and the cycle will repeat itself. Here, for the carbon dioxide, every 100 ml of blood leaving the tissue carries with it 4 ml of carbon dioxide to the lung, and the cycle will repeat itself on and on. Unlike the oxygen, which is transported mainly by the hemoglobin, the carbon dioxide is transported by 3 methods. The first method is the transport of carbon dioxide in the dissolved state. Please remember that the carbon dioxide is highly soluble in water, and much amount of the carbon dioxide leaving the tissues can dissolve simply in the plasma. This method of transportation accounts for not more than 7% of the transportation method. The second transportation method is the transport of carbon dioxide in the form of bicarbonate. This is the most important method of transportation, and this accounts for about 70% or even more for the transportation of carbon dioxide. And the last method of transportation is the transportation of carbon dioxide in combination with hemoglobin and plasma protein. And this accounts for about 20 to 23% for the transport of carbon dioxide. Now we need to discuss each method of transportation separately. Transport of carbon dioxide in the dissolved state. We mentioned that the carbon dioxide is highly water soluble, and the amount of carbon dioxide dissolved in the fluid of the venous blood at 45 millimeter of mercury is about 2.7 ml per deciliter. As 2.4 ml per deciliter of carbon dioxide is dissolved in the arterial blood, this means that the arterial blood carries carbon dioxide in the dissolved state, and the venous blood carries carbon dioxide in the dissolved state. But the concentration of the carbon dioxide in the venous blood in the dissolved state is higher than that in the arterial blood. The difference between these two concentrations are only 0.3 ml per deciliter of carbon dioxide. And this means that 0.3 ml per deciliter of carbon dioxide is transported from the venous blood to the lung. If you take the percentage of this 0.3 ml per deciliter out of the 4 ml per deciliter that is carried to the lung in every cycle, this will be about 7% for all the carbon dioxide normally transported. Please remember that this is a minor method for the transportation of carbon dioxide. The transport of carbon dioxide in the form of bicarbonate ion. Please refer to this figure. The figure will explain this mechanism for you. As we know that metabolism produces carbon dioxide within the cells in the peripheral tissue. Then the carbon dioxide, by simple diffusion, will leave the cell out to the interstitial space. And then from the interstitial space, it will get into the blood that's within the capillaries supplying that tissue. Once the carbon dioxide is within the blood, it will diffuse by simple diffusion again inside red blood cells. Inside red blood cells, carbon dioxide will react with water to form carbonic acid. This reaction is catalyzed by the huge amount of carbonic anhydrase enzyme present within red blood cells. This phenomenon allows tremendous amount of carbon dioxide to react within the red blood cells with water even before the blood leaves the tissue capillaries. The reaction is so huge and so fast, and this enzyme can increase the rate of production of the carbonic acid by more than 5,000 times. As you know, carbonic acid further dissociates into hydrogen ion and bicarbonate ion. Most of the hydrogen ions then combine with the hemoglobin. I hope you know that hemoglobin is a protein with a negative charge, and this will attract the hydrogen ion, and that's why we consider hemoglobin as the most important intracellular buffer. Now, for the huge amount of bicarbonate that has been produced within the red blood cells, many of the bicarbonate ions diffuses out from the red blood cells into the plasma. And for keeping electricity equal across the cell membrane, for each bicarbonate ion leaving the cells, one negative chloride ion diffuses into the red blood cells to take their place. This phenomenon is called the chloride shift. So please remember, at the tissue site, huge amount of a chloride ion gets inside red blood cells. And the contrary will happen when the blood reaches the lung. There, chloride ion will diffuse out of red blood cells and the bicarbonate ion gets in, as one will see soon later. The third method of the transport of carbon dioxide is the transport of carbon dioxide in combination with hemoglobin and the plasma protein. Carbon dioxide reacts directly with a loose bond with amine radicals of the hemoglobin molecule to form the compound carbaminohemoglobin, and it can be transported as such, attached to the hemoglobin. A much smaller amount of carbon dioxide also react in the same way with the plasma proteins in the tissue capillaries. However, this reaction is less significant than hemoglobin transport of carbon dioxide. The student should know that the contribution of carbaminohemoglobin and plasma protein in the transport of carbon dioxide to the lung is just above 20% of the total quantity transported. Please note that the loose bond between the hemoglobin and the carbon dioxide, although it's loose and can release carbon dioxide from the hemoglobin in the lung, still it is not as fast as the release of carbon dioxide in the lung from the union of the carbonic acid with the union of the bicarbonate ion with the hydrogen ion to reform the carbonic acid and releasing the carbon dioxide in the lung. So this method of transportation is much less efficient than that of the bicarbonate method. This figure shows you the carbon dioxide dissociation curve. It is not like that of the oxygen dissociation curve, which is sigmoid in shape. And please note this most important difference between the two curves. The normal blood carbon dioxide range is very narrow here, not as in the oxygen dissociation curve, which has a wide range of action between partial pressure of oxygen of 40 in the venous blood and partial pressure of 97 millimeter mercury in the arterial blood. Here the range for the carbon dioxide is between the value of 40 and 45 millimeter mercury. So it is a narrow range. Please note that the concentration of the carbon dioxide rises to about 52 volumes percent as the blood passes through the tissue and falls to about 48 volumes percent as it passes through the lung. Therefore, only 4 volumes percent of the carbon dioxide concentration is exchanged during normal transport of carbon dioxide from the tissue to the lung. The Haldane effect. The Haldane effect is a property of hemoglobin first described by Professor John Haldane. He noticed that oxygenation of blood in the lung displaces carbon dioxide from hemoglobin, which increases the removal of carbon dioxide. So this property has been named as the Haldane effect. That's to say, binding of oxygen with hemoglobin tends to displace carbon dioxide from the blood. The explanation for this effect is that the combination of oxygen with hemoglobin in the lungs causes the hemoglobin to become a stronger acid. The more highly acidic hemoglobin has less tendency to combine with carbon dioxide to form carbaminohemoglobin, thus displacing much of the carbon dioxide that is present in the carbaminohemoglobin from the blood. Please take a look at the graph at this figure, and this will explain to you the physiological significance of the Haldane effect. In this graph, we see portion of the carbon dioxide dissociation curve. The red solid line is the carbon dissociation curve of the arterial blood, and the dotted line is the carbon dissociation curve of the venous blood. Please take a look at point B. At point B, it shows you how much the blood is saturated with carbon dioxide in the arterial blood, where the partial pressure of carbon dioxide is 40. It's about, as you can see, it's about 48 by volume, means 48 mils of carbon dioxide carried by each 100 ml of blood. Now, if this blood reaches to the tissues and exchange took place, and the blood will be converted to the venous blood without the Haldane effect, at the pressure of 45 millimeter mercury, the concentration of the carbon dioxide should be 50. Please look at the arrows I made. But in reality, Professor Haldane discovered that at the partial pressure of 45 in the venous blood, the blood will hold a concentration of carbon dioxide equals to 52. So there is an increase in the carbon dioxide held by the hemoglobin, and this is simply because the hemoglobin has been changed from oxygenated hemoglobin to deoxygenated hemoglobin, and this is the Haldane effect. Simply, oxygenated hemoglobin holds less carbon dioxide than deoxygenated hemoglobin. This is important not only in the tissue, but it is important in the lung, because when the venous blood turn into blood returning to the heart and the blood pumped by the right ventricle to the lung, when the deoxygenated blood reaches the lung, and the lung will get rid of the carbon dioxide and more oxygen will be attached in the hemoglobin will liberate more carbon dioxide. So the Haldane effect simply doubles the amount of the carbon dioxide released from the blood in the lungs, and approximately doubles the pickup of carbon dioxide in the tissue. Without the Haldane effect, the transport will be just 2 mils of carbon dioxide for every 100 ml of blood. There is another minor effect within the Haldane effect, that is, the increased acidity of the hemoglobin will increase the release of the hydrogen ion, and by increasing the hydrogen ion, this will increase the hydrogen ion available to be reunited with the bicarbonate ion to form carbonic acid, and carbonic acid will be dissociated into water and CO2, and more CO2 will be released, and this will end into the alveoli. So you can see that the Haldane effect affects mainly the attachment of the carbon dioxide to the hemoglobin, and the minor effect is by enhancing the release of the carbon dioxide from the bicarbonate in the lung. I hope you understand well this lecture. Will be, as usual, the test question I put for you, and please note that this question is about the arterial blood, not about the venous blood. Try to solve it, and if you have a difficulty in solving it, or if you have any question about it, please don't be hesitant to ask me directly or by the email. And thank you.